Now, let us look into the different sampling techniques for viruses. Selection of viral sampling site depends on the virus type, the suspected site of infection, and the test requirements. Common techniques for viral sampling include collection of swabs. For respiratory viruses, nasopharyngeal swabs are used for viral sampling. For sexually transmitted viruses, genital swabs are obtained. And for viruses that grow on surfaces, environmental swabs are used for sampling. Blood drawing. This helps in detecting viruses present in the bloodstream, often used for HIV or hepatitis B and C viruses. Tissue biopsy obtains a small tissue sample for viruses that target specific organs. For example, liver biopsy for hepatitis B virus. Airborne virus sampling collects airborne viral particles for studying transmission patterns and assessing environmental contamination. Example, COVID-19 virus. Virus isolation and cultivation. Viruses are obligate intracellular parasites, so they depend on host for their survival. They cannot be grown in non-living culture media or on agar plates alone. They require living cells to support their replication. Viruses can be grown in vivo within a living organism like a plant or an animal, or in vitro outside the living organism in cells in an artificial environment such as a test tube, cell culture flask, or agar plate. Why do we need to cultivate viruses? To isolate and identify viruses in clinical samples, to prepare vaccines and antigens against viruses, to study viral gene expression and viral replication, also to decipher viral structure and to understand viral genetics in a better way, to study the pathogenesis of viral diseases and viral oncogenesis, that is, to study the effects of viruses on host cells. Now, let us look into one of the methods of cultivating viruses, that is, by inoculation into embryonated eggs. The process of cultivation of virus in embryonated egg depends on the type of the egg which is used. Viruses are inoculated into chick embryo that is 7 to 12 days old. For inoculation, eggs are first prepared by cultivation. The shell surface is disinfected with iodine and penetrated with a small sterile drill. After inoculation, the opening is sealed with gelatin or paraffin and incubated at 37 degrees centigrade for 2 to 4 days. After incubation, the egg is broken and the virus is isolated from the tissue of the egg. Viral growth and multiplication in egg embryo are indicated by the death of the embryo, by embryo cell damage, or by formation of typical pox or lesions on the egg membranes. Viruses can be cultivated in various parts of the egg, like the chorioallantoic membrane, the allantoic cavity, the amniotic cavity, or the yolk sac. Advantages of inoculation into embryonated eggs. This is a widely used method for isolation of virus, for studying viral growth, and also for vaccine production. Eggs are available in unlimited amounts. Eggs are easy to handle. They provide a sterile environment free from any contaminating bacteria and many latent viruses. They are not able to produce antibodies. Disadvantages of inoculation into embryonated eggs. The site of inoculation varies for different viruses. Different viruses have different sites for their growth and replication. Therefore, Sufficient skill is required for inoculation into embryonated eggs. Animal inoculation is yet another method for cultivation of viruses. Viruses that cannot be cultivated in embryonated eggs or tissue cultures can be cultivated in laboratory animals such as mice, guinea pig, hamster, rabbits, and primates. The selected animal should be healthy and free from any communicable diseases. Mice are the most widely used animals in virology. Mice can be inoculated through several routes such as intracerebral, subcutaneous, intraperitoneal, and intranasal. Mice are usually inoculated with the challenge virus and observed for 21 days. They can be studied by collection of oral swabs or by euthanizing the animals, dissecting them, and sampling of various organs such as the brain, salivary gland, spinal cord, head, and hind legs. Advantages of animal inoculation: diagnosis, pathogenesis, and clinical symptoms associated with viruses can be easily determined. Production of antibodies in animal models can also be identified. These models are suitable for primary isolation of certain viruses. Mice provide a reliable model for studying viral replication. They can be used to study immune responses, epidemiology, and also oncogenesis by certain viruses. The disadvantages of animal inoculation: animals can be expensive and there are certain difficulties in maintenance of animals. It is difficult for choosing animals which suit for a particular virus. Some human viruses cannot be grown in animals or can be grown but do not cause disease. Mice do not provide models for vaccine development. It can also lead to the generation of escape mutants. Further, there are several issues related to animal welfare systems. Cell culture based techniques. Tissue culture was first performed in the early 1900s for studying animal cells free of variations caused by homeostasis in the whole animal. For the first 50 years of tissue culture, the technique was performed with fragments of explanted tissue. So the term tissue culture has stuck in spite of the fact that since 1950s, dispersed cells in culture or cell culture has been practiced. Therefore, tissue culture sometimes refers to cultures derived from dispersed cells taken from the original tissue, from primary culture, or from a cell line or cell strain. In the 1950s, it was discovered that human tumor cells could give rise to continuous cell lines. Example, HeLa cells. There are three types of tissue culture: organ culture, explant culture, and cell culture. Organ cultures are mainly done for highly specialized parasites of certain organs. For example, the tracheal ring of the alpaca is used for the isolation of the Middle East respiratory syndrome coronavirus, the MERS-CoV virus. Here, the tracheal tissue is placed on a gauze placed in a culture media and it is inoculated with the virus. Further, it is sampled for every 24 hours for up to 72 hours. Explant culture, however, is rarely done for viral sampling. Culture-based techniques. Cell culture is mostly used for the identification and cultivation of viruses. Cell culture is a process by which cells are grown under controlled conditions. Cells are grown in vitro on glass or treated plastic surface in a suitable growth medium. At first, the growth medium, a usually balanced salt solution containing amino acids, sugar, protein, salts, carb serum, buffer, antibiotics, and phenol red are taken, and the host tissue or cell is inoculated. On incubation, the cell divide and spread out on glass surface to form a confluent monolayer. Incubation of cell culture tubes in a stationary rack can be used in place of a roller drum. To counteract the pH decrease, a bicarbonate buffering system is used in the culture medium to keep the cells at physiological pH 7.2. Phenol red, a pH indicator, that is red at physiological pH, yellow at acidic, and purple at alkaline pH. Once inoculated with the specimen, the cells are incubated for 1 to 4 weeks depending on the viruses involved. Periodically, the cells are inspected microscopically for the presence of the virus, indicated by areas of dead or dying cells, also called as the cytopathic effect. Two kinds of media are used in cell cultures. One is the growth medium, as already mentioned, and second one is the maintenance medium. Usually, antibiotics such as vancomycin at 10 mg per ml, gentamicin at the concentration of 20 mg per ml, and amphotericin at the concentration of 2.5 mg per ml are added to avoid any bacterial growth or contamination. A cell culture becomes a cell line once it has been passed or subcultured in vitro. Primary cell cultures. These are normal cells derived from animal or human cells. They are able to grow only for a limited time and cannot be maintained in serial culture. That is, only 5 to 10 passages in culture. They are used for the primary isolation of viruses and production of vaccine. Examples include monkey kidney cell lines, human amnion cell lines, and heterogeneous population of cells. Diploid cell lines or semi-continuous cell lines. These are also derived from normal cells of animals or humans. They can be subcultured up to 50 to 100 passages by serial transfer following senescence, and the cell strain is then lost. They are used for the isolation of some fastidious viruses and the production of viral vaccines. Examples include human embryonic lung strain, rhesus embryo cell strain, and homogeneous population of cells. Continuous cell lines or heteroploid cell lines. They are derived from cancerous cells. They can be serially cultured indefinitely, so are named as continuous cell lines. They can be maintained either by serial subculture or by storing in deep freezer at -70 degrees centigrade. Due to derivation from cancer cells, they are not useful for vaccine production, while its importance lies in the identification and cultivation of viruses. Examples include the HeLa cell line, the human carcinoma of cervix cell line, the HEP2, that is the human epithelioma of larynx cell line, the vervet monkey cell lines, kidney cell lines, BHK21, the baby hamster kidney cell line. Detection of viral growth in cell culture. Multiplication of virus can be monitored in a variety of ways. Development of cytopathic effects or other mean morphological microscopic changes that occur in virus infected cells. The type of virus induced cytopathic effects include cell lysis, necrosis, apoptosis, formation of syncytia, or cytoskeletal disruption. Certain viruses, like enteroviruses, could also form inclusion bodies. The inclusion bodies can be nuclear, as in case of cytomegalovirus, or it can be cytoplasmic, as in case of reovirus. These are some of the cytopathic effects seen in viral infected cells. This is the appearance of healthy cells under the microscope. Here we can see the rounding of cells in infected cells. Also, there is formation of giant cells called syncytia. Certain viruses induce the formation of plaques. Vacuolation can also be seen in some cells. Some others show the formation of intracytoplasmic inclusion bodies. The advantages of cell culture is that it is relatively easy to perform and it can be performed for cultivation of a broad spectrum of viruses. It is cheaper and more sensitive. Disadvantages of cell culture. The process requires trained technicians with experience in working with cell cultures on a full-time basis. State health laboratories and hospital laboratories do not isolate and identify new viruses in clinical work. The tissue or serum for analysis has to be sent to central laboratories for viral identification. Precautions to be taken while sampling viruses and culturing them. Sampling viruses requires careful attention to prevent contamination and ensure accurate results. Here are some precautions to take. Use personal protective equipments like personal protective equipments which include goggles, face shield, medical cap, surgical mask, N95 mask, shoes cover, alcohol gel or alcohol spray, disposable gloves and leg cover. To protect yourself from potential exposure to the virus, work in a biosafety cabinet. These provide controlled environment to minimize the risk of exposure and contamination. Sterilize equipment. Autoclave or disinfect all equipment including pipettes, tubes and other tools before and after use to prevent cross-contamination. Follow standard operating procedures. Adhere strictly to established protocols for sampling, handling and disposing of viral samples. Minimize aerosols. Use techniques that minimize the generation of aerosols, such as pipetting slowly and carefully and avoiding vigorous shaking or vortexing. Avoid splashes and spills. Handle samples with care to prevent spills or splashes. Clean up any spills immediately using appropriate disinfectants. Label samples clearly. Clearly label all samples with the date, time, and type of virus to avoid confusion and cross-contamination. Dispose of waste properly. Dispose of all biohazardous waste, including used personal protective equipment and contaminated materials according to local regulations and institutional guidelines. Clean work surfaces regularly. Disinfect work surfaces before and after working with viral samples to prevent contamination. Practice good hand hygiene. Wash your hands thoroughly with soap and water before and after handling viral samples and use a hand sanitizer as needed. Monitor for symptoms. Be vigilant of any symptoms of illness and report them promptly to the appropriate authorities if you suspect exposure to a virus. Biosafety and biosafety levels. What is biosafety? Biosafety is the application of safety precautions that reduce a laboratory's risk of exposure to potentially infectious microbe and limit contamination of the work environment and ultimately the community. What are biosafety levels? There are four biosafety levels. Each level has specific controls for containment of microbes and biological agents. The primary risks that determine the levels of containment are infectivity, severity of disease, transmissibility, and the nature of work conducted. Origin of the microbe or the agent in question and the route of exposure are also important. Each biosafety level has its own specific containment controls that are required for the following: laboratory practices, safety equipment, facility construction. Biosafety levels range from biosafety level 1 to biosafety level 4. Each biosafety level builds on the controls of the level before it. Every microbiology laboratory, regardless of the biosafety level, follows standard microbiological procedures. Now, let us look into the various biosafety levels and its significance. This figure describes some of the measurements taken when practicing biosafety level 1 and biosafety level 2. The figure shows an example of the agent that requires biosafety level and the personal protective equipment primary that are generally used when performing laboratory work with that agent. The right side of the figure shows the secondary precautions that include facilities construction and engineering. The color of the biohazard sign indicates the level of danger and biocontainment. Biosafety level 1 controls microorganisms usually known to cause mild disease or disease with minimal hazards to the laboratory and community. Biohazard level 1 usually includes viruses that cause chickenpox and many non-infectious bacteria like E. coli. The level of precaution at this level is minimal and usually involves wearing a face mask and no close contact. Safety protocols for biosafety level 1 labs, which require only standard microbial practices, include mechanical pipetting, that is, no mouth pipetting is allowed, safe sharps handling, avoidance of splashes or aerosols, daily decontamination of all work surfaces when work is complete, regular hand washing, prohibition of food, drink, and smoking materials, the use of personal protective equipment such as goggles, gloves, and the lab coats or a gown, and sticking biosafety hazard signs throughout the laboratory. Now, the biosafety level 2. Biosafety level 2 controls microorganisms generating moderate hazards to the laboratory and the community. Biosafety level 2 involves microorganisms that are responsible for mild infections in humans and are often difficult to contract via aerosolized particles like hepatitis A, B, and C, Lyme disease, salmonella, measles, mumps, HIV, and dengue. In addition to the safety protocols established for the biosafety level 1, biosafety level 2 labs are subject to the following safety controls: the use of PPE, including lab coats, gloves, eye protection, and in some cases, face shields. All procedures that could cause infection from aerosols or splashes must be performed with the biological safety cabinet. Decontamination of infectious materials prior to disposal, generally through the use of an autoclave, self-closing lockable doors, access to a sink and eye wash station. Biohazard warning signs should be placed throughout the laboratory. Secondary barriers include self-closing doors, installed biosafety cabinet tool, heat and chemical resistant benches, eye wash stations. Windows are not recommended. Cleanable furnitures. Biosafety level 3 includes the control of infectious agents which cause both serious hazards and can cause a potentially lethal condition to the laboratory and community via respiratory transmission of the organism. Biohazard level 3 includes microorganisms that can be fatal to humans, but for which vaccines are available. Aside from Mycobacterium tuberculosis, this grouping includes anthrax and many types of viruses causing encephalitis, hantavirus, Rift Valley fever, Rocky Mountain spotted fever, and yellow fever virus. Common safety controls within biosafety level 3 laboratory include the use of PPE, including goggles and gloves. Respirators may also be required. The use of solid front wraparound gowns, scrub suits, and or coveralls is often required. Access to hands-free sink and eye wash station available near the exit. Sustained directional airflow to draw air into the laboratory from clean areas toward potentially contaminating areas. Exhaust air cannot be recirculated. Self-closing set of locking doors which with access away from general building corridors. The secondary barriers facilities include partially isolated laboratory, double pass doors, ante-room for clothing change, self-closing doors with locks, air ventilation system which is ducted, filters protecting vacuum lines, class 3 biosafety cabinet, decontamination tools for waste, cleanable walls and ceilings, and sealed windows. Biosafety level 4 is the highest and the most complex biohazard level involving a relatively few clinical microbiology laboratories. Biosafety level 4 labs are extremely isolated, often located in an isolated and restricted zone of a building or in a separate building entirely. Biosafety level 4 labs also feature a dedicated supply of exhaust air, such as vacuum lines and decontamination systems. Biohazard level 4 usually includes dangerous viruses like Ebola, Marburg virus, Lassa fever virus, Bolivian hemorrhagic fever, and many other hemorrhagic viruses found in the tropics like the Crimean Congo virus. In addition to biosafety level 3 considerations, biosafety level 4 laboratories must follow these safety protocols. Personnel must change clothing before entering the facility and shower upon exiting. All materials must be decontaminated before leaving the facility. Personnel must wear PPE from lower biosafety levels as well as a full body air supplied positive pressure suit. Access to Class 3 biological safety cabinet. The secondary barriers facilities include isolated laboratory, installed biosafety level 3 cabinet, outer and inner clothing change areas separated by shower room, unbreakable and sealed windows, chemical shower, suit decontamination, emergency power source, communication systems, microphone, non-recirculating ventilation system. Now let us look into few MCQs. Which biosafety level is recommended for handling highly infectious viruses? A. Biosafety level 1 or BSL-1. B. Biosafety level 2 BSL-2. C. Biosafety level 3 BSL-3. D. Biosafety level 4 BSL-4. The answer is D. Biosafety level 4. Which of the following is not a recommended precaution during viral isolation? A. Using personal protective equipment PPE. B. Working in a biosafety cabinet. C. Using disposable equipment. D. Eating or drinking in the laboratory. Answer is D. Eating or drinking in the laboratory. Which of the following is not a recommended practice for biosafety during viral culture? A. Proper decontamination of all waste materials. B. Regular monitoring of laboratory personnel for symptoms of viral infection. C. Sharing personal protective equipment among laboratory members. D. Implementing strict access controls to the laboratory. The correct answer is C. Sharing personal protective equipment among lab members. these are a few of the references. Thank you for listening.